So thank you for joining us, everyone, here today. So a brief overview of what we're going to be covering. At any point, go ahead and put questions in the Q&A. We'll save most of them to the end, or Carol and Jeremy will be answering them by typing out answers. And we'll post all of the Q&A and answer any leftover questions that we don't get to in today's session on our community forum afterwards. And you can also post follow-up questions there. So then we'll start with the Ellen Developing Mouse Brain Atlas with Carol, covering the development of this resource, how to access it, and some use cases. Why is this a powerful tool for your work? And then move into the BrainSpan Atlas of the developing human brain. And again, review the resource development, access, and some use cases with Jeremy. And again, we'll have Q&A throughout. As a reminder, this tutorial is the first in a series that will be running monthly through February that highlights selected open resources from the Allen Institute for Brain Science. And it will culminate in a symposium and more tutorials on March 8th through 10th, 2021. We'll hope you join us for more of these upcoming events, more of these tutorials, and for the symposium in March. The links to register for those, if you have not already, are going to be in the chat. So again, please place all questions for the panelists in that Q&A panel on Zoom, or you can ask them in the YouTube chat. Use that Zoom chat to talk to each other about your research interests, share observations, and other comments. And please make sure you're sending all your chat messages to all panelists and attendees so others can see your message. We do have closed captions available. There is a link to click in the chat. Our speakers today are Carol Thompson, who will be presenting on the Allen Developing Mouse Brain Atlas, and Jeremy Miller, speaking on the BrainSpan Atlas of the developing human brain. Both of them were involved in the development of the atlases they'll be sharing with you today. So I'm excited to hear those presentations. The Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, cell biology, and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental biological sciences. And our approach is guided by 3 principles, which I'll return to in a moment. Our work generates data, knowledge, and analysis tools, all of which we share publicly. To date, we've collected about 8 petabytes of data. That's 1.7 million DVDs worth that we have shared with the public. So those 3 core principles are team science, big science, and open science. And the most relevant today is open science, which is that we share all of our data and all of our data analysis tools for you to use in your work. Please check out some of the other webinars in the series to learn more about those open resources, or check out all of them on brain-map.org. Today, we're going to be talking about our atlases of the developing brain, the Allen Developing Mouse Brain Atlas and the BrainSpan Atlas of the developing human brain. The mammalian brain progresses through a consistent series of stages over the course of development, much of which is conserved across mammalian species. Steps include establishing the axes or directions of the brain, neuronal generation and migration to form those cortical layers, and continuing development postnatally. These stages of development are linked to transient and patterned gene expression, which is what is captured in these atlases. Multiple transient layers of progenitor cells will appear during development, some of which eventually become neurons or glia, and that we see excitatory neurons generating starting with deep layers and working outwards, while inhibitory neurons migrate in from other structures. The mouse and human brain analysis we're going to show you today enables users like you to explore gene expression across the developing mouse and human brain at multiple stages of development, capturing some of these significant developmental events. You might want to use this data in conjunction with your own, for example, to look up the expression pattern of your gene of interest across multiple stages of development, or as the main focus of your analysis. Carol and Jeremy are going to demonstrate for you where to find this data and some example use cases of how and why it can be applied. One last reminder, please use the chat to chat with each other and introduce yourself while the presentation is ongoing, and questions should end up in that Q&A panel. All right, Carol, I'm going to turn it over to you.
Hello, my name's Carol Thompson. Let me try to share my screen here for a moment. Lost my window there for a second. Okay. Thank you very much for that introduction, Caitlin, and for introducing a little bit about the brain and development. I am very excited to be able to share with you today our Allen Developing Mouse Brain Atlas that I had the pleasure of getting to develop some years back. So the initial Allen Institute product that we developed in 2003 to 2006 was the Allen Mouse Brain Atlas. This atlas profiled the expression pattern of about 20,000 genes in adult mouse brain using a technique called in situ hybridization, or ISH. This technique shows the expression of individual RNAs probed with RNA probes, all in the context of brain slices. You can really see where the gene expression is relative to the anatomy. The atlas that we put up with this was a reference atlas that was drawn by Hongwei Dong. And this atlas and the gene expression data were all mapped to a 3D space, a common coordinate framework. And having both data mapped in and the reference atlas gave us the ability to create a number of different tools for search and navigation and mining of the data. The Allen Developing Mouse Brain Atlas was a follow-on project with the goal of using the same methodologies to be able to extend our understanding of gene expression in the mouse brain by using these same methods to look at expression across 4 embryonic and 3 postnatal ages. So this work was largely done from 2008 to 2010, although we had some updates past 2010 as well. So the brain's an extremely complicated organ that functions much like a computer, but as we are not the designers of the system, we have to reverse engineer the brain to understand how it's wired, what are the component parts, and in order to be able to develop therapies or think about how we counter various disorders, we need to be able to understand this. So a major goal of many of the studies that have been going on over the last decade or so is the creation of a parts list of cell types and connections so that we can basically have an inventory of what is there to help us understand how things might function. Transcriptomics, or the study of gene expression in the brain, is one way that we can get a handle on identifying and understanding cell types. Different cell types are marked by different genes that are highly expressed, and these genes underlie proteins that give those individual cell types their unique functions. While many are using transcriptomics to profile individual cells in adult brain, neuron identities and their connectivity and function are influenced by their position in the brain and the signals they receive during development. And in fact, the neurons move through development sometimes throughout the brain before arriving in their final position. These dynamics in transcriptomic signatures, as we can see even in hippocampus, you can look at a gene from the embryonic E13.5 age all the way up to near adult at P28. You can see expression will mark specific regions, like here, the CA region of the hippocampus. But we can also see that there's dynamics and nuance to that expression such that something may shift from being expressed in the entire region to being expressed only in CA1 or CA2, and sometimes the regionality of these expression patterns changes completely. So having a reference set of gene expression in an anatomic context or in situ like this is important for those who are seeking to study model systems if they are trying to create neurons that represent development in model systems like organoids, or to manipulate other in vitro systems. They need a reference to the actual animal and what those gene expression signatures might be, what those cell types might be. And that's why these developmental atlases are a really great reference for all sorts of other studies for understanding adult brain function and developmental disorders. So we created the Allen Developing Mouse Brain Atlas. It's a lot of work to do the in situ hybridization. So we expanded the number of time points that we had to do with 4 embryonic and 3 postnatal time points. But we limited it to a set of 2,100 important genes with families that would be highly relevant to development, brain disorders, or as markers for particular cell types or signaling pathways. And again, we had reference atlases drawn. We used Luis Pueyas from the University of Murcia, hand drew a number of these reference atlases at all these different time points. And our overall goal for bringing all this together is to profile genes relevant to development or developmental disorders, be able to access and understand the dynamics of gene expression, and provide easy search and navigation to enable discovery of new genes with important roles in brain development. So all the in situ hybridization experiments mean that we have individual sections of tissue that are sliced. Each one is sliced and then probed for individual genes. So every slice shows you where a particular gene might be expressed across that anatomy. And while histology is great with thousands of genes, many thousands of experiments and images, informatics tools are needed to help users actually be able to find the data they're interested in. So these reference atlases were not only hand drawn, we also then lofted them into a 3D context so that we could create tools to map everything into one 3D framework and allow us to develop some of the tools I'm about to tell you about. So some of the search tools that we created, basically every experiment's worth of in situ hybridization data, every slice was registered into a 3D space, and then the expression signal was segmented out, and we can summarize it across the brain. If you take the brain and divide it into a series of voxels or cubes, so you cube the whole brain, and we map that expression into these cubes, we can do a lot of summaries of expression by voxel in the brain, and we can do a lot of correlation measurements. So two of the tools that we have, one was our correlation search, originally called Neuroblast. This is one where basically if you were to say, look at the diencephalon of the brain and take those voxels, you could make a vector of voxels. So vectors of pieces of the brain in space, and you could have measurements of that gene expression in that vector. And you could see how gene 1 correlates to gene 2 correlates to gene 3, and find which genes are most highly similar using a Pearson's correlation. This gets you genes, we like to call it a genes like me. Find genes like me. I've got an interesting gene. I want to find other genes that look just like this. The other tool that we have is our anatomic gene expression atlas. This one's a little more complicated. Basically, what we do here is we say, take the entire data set, 2,100 genes. We make a vector of the genes. So for every gene, for every voxel, we can say, now let's compare that vector of genes for this voxel versus that voxel versus that voxel. And we can see how correlated different voxels in the anatomic space of the brain, how correlated they are. And in that way, you can see voxels that have similar expression patterns to each other. So this is basically letting the gene expression tell you which parts of the brain are related at a transcriptomic level. So I'll show some of these to you in the context of the website here. I just want to point out this will be the landing page of the website. Today, I'm going to show you a little bit about how you do gene search and get into the anatomy. If I have time, I will briefly show you the anatomic and temporal search. And I'm also going to show you the AGEA. The neuroblast or correlation search, I will show you in the context of the gene search here. Okay. So some example use cases you might walk into this resource with, you might have different questions about the brain or about development or about genes. One would be, well, where are Wnt signaling pathways active during development? You might also ask, can we identify candidate transcription factor networks based upon the co-expression of transcription factors? And finally, we might ask, what genes are active in the thalamus in development or any other region that you look at? With that, I'm going to open up the website and let's walk through this a bit. Okay, so the homepage, I'm starting here at our portal to brain-map.org. And to get to the Developing Mouse Brain Atlas, you can either scroll down and find it here, or you can go up to the menu up here and come down and click Developing Mouse Brain Atlas. So this takes you to the homepage for this resource. Here, you can start off by searching for the gene of interest, or you could browse by and get a gene list associated with one of these gene classifications. So you could go straight to Wnt signaling pathways and pull up a whole list of Wnt signaling. But I'm going to start off here by going with, let's look for this transcription factor, POU4F1. And this brings back, we only have one gene in that matches that gene search here. And what we have is a little expression summary here. I'll show you this a little bit more in the context of the expression page. So if I click on this, and I'm going to open it in a new tab, I clicked on the gene symbol, and it's going to open up the gene detail page for me. And so here we have the name of the gene, POU domain, class 4, transcription factor 1, aliases, entree ID number, chromosome, and organism. And you have the expression summary. So this is the expression of this gene quantified in that 3D space and summarized up to kind of high level anatomic regions up here. So the thalamus is basically in the prosencephalon here, these P1, 2, and 3. I think mostly P1, P2 has our thalamus in it. And then we have the forebrain over here, and we have the hindbrain, arranged anterior to posterior. Red means higher expression. So you can see that a lot of these genes are expressed mostly from the thalamus onto the hindbrain in early development. Now you can look down here and see an example image from each experiment. This particular gene has both coronal and sagittal experiments available. Here's a coronal, here's a sagittal, and we have 3D thumbnails you can look at that summarize the expression in 3D for that particular experiment. So if I rotate this, you can see where the expression is. So I could go directly from here to opening up individual experiments, but I am going to go back this page and show you if I click on this row here, I get the same 3D thumbnails over here. So anytime you want to look at a gene, you want to see where it's expressed at a glance in 3D across all the time points. You can also come over here and do the same trick of rotating all the thumbnails and seeing if there's expression at each time point that you're interested in. So what I'm going to do here is select some experiments to take a look at. And to do that, you use these check boxes that are right next to the expression. So I'm going to go click, and I could have done this from the gene detail page as well, but I'm going to work from here. Actually, I'm just going to limit this to 3 embryonic ages to make it easy. There's no limit to how many genes you can put into your cart. It will tell you how many genes you have in your cart down here. I'm going to hit view selections. I like to mine data. I like to load 20, 30, 50 genes in the browser at a time, and I like to go mine through and see which experiments are interesting. And what's nice is you can completely tailor your view to how you want to do it. So it pre-opened right now, and it defaulted to 2 columns. But I know I have 3 experiments, so maybe I want to see 3 columns. I did that rather fast, but there's a little settings setting icon up here in the upper right. So you can click that to choose how many columns you want for your page. So I set it to 3 because I have 3 experiments I'm going to look at. And then, you know, it would be really nice to have some reference atlases to look at the same time. So I clicked this atlases drop down here, and I'm going to select the atlases that correspond to these time points for developing mouse, and close, and they will pop up as well. And if you don't like the order of how these things are, I am always one, I like my gene expression at the top. I like to line up my E11.5s are here. I'm going to put my E13.5 ISH there and the atlas here. And there we go. This is how I want to look at this experiment. Then another thing you could do is there are zoom and pan controls on the window, but I just like to use my mouse. So I'm using the roller wheel to zoom in. And then to go forward one section at a time, you can either click down here, or once you're zoomed in, you can hit F. And it will maintain the same zoom as you click F to go forward. You would click D to go back a section, but F to go forward. So you could just keep going through, or you can shortcut and take it a little further in. What's interesting then is if I looked at one experiment and said, hey, look, there's interesting expression here in the midbrain at E11.5. If you want all the other windows to sync to that window so that you're looking at the same part of the brain across all the time points, you click S. There's also, I believe you can use this icon right here at the top of your window to do a sync. And voila, all of your images will sync up. You could have 50 experiments in here and do that sync as well. And that is one of the handiest tools that we have. So there you can see that great expression across all these different windows. It synced up the reference atlases. If you were wondering, what is this part of the brain? You could just say, well, let's go to the reference atlas. In that window, you click key. There's a key symbol right here. And if you open that up, you can have a browsable window to look at the reference atlas with its ontologies. And again, you can navigate in the view much like you do in the other window. You can zoom in with the roller wheel, And then you can also do a search if you wanted to search for a structure. So if I wanted to know what SC was, I could type in the search window at the top, SC, and I could find, oh, that's superior calendula. And you can also, if you click on a region in the Atlas ontology on the left, it will highlight for you in purple that same region of the Atlas. And the same, if you click over here, it will show you where that structure is with its full structure name in the reference Atlas ontology. Okay, so it's a really nice way to be able to go look at expression in the brain. And here I'm looking and I'm seeing, hey, there's some interesting expression right here in part of the thalamus at E13.5. So I might want to go back and take a look at that. So here's one of our tools. If you click on this gene, where these 3D models come up on the right, you have your ability to do a correlation search to find genes like me. So if I want to find genes that look like POU4F1, I would say I would limit my search to which structure of the brain I wanted to look at. So maybe here I wanted to look at the diencephalon. And I think I'm going to search this one at E15.5. And so let's go see what other genes look like this transcription factor E15.5. So I click search and you come back with a list of genes. If you want to see more, it's only showing 3 by default, but you can choose to look at 25 genes by clicking this other little settings icon here and clicking how many genes you want to see. And then you can scroll through them in the window. And if I look through, they are rank ordered by their correlation to my seed gene. My seed gene was POU4F1, searching at E15.5 in the diencephalon. And so when I look through, I see a lot of other transcription factors coming up too. IRX2 is a transcription factor. If I go down further, I have ETV1 and NECAB2. And what's interesting is if I were studying this gene, maybe I would know that there's a paper that had been published that showed that in a POU4F1 knockout mouse, that a number of these transcription factors were actually shown to be downregulated. So it's really interesting to see, oh, these were co-expressed with this one, and we have reason to believe they might be downstream in a transcriptional regulatory cascade of the POU4F1. So in general, this might be an interesting way to find co-expressed transcription factors and have a narrowed candidate list of genes that might be acting in the same regions and be under control of the transcription factor. Just a candidate list, you can't, you would need to do more work to confirm that. Okay. So if I were to do the other case and I put in Wnt3A, so maybe I'm interested in Wnt signaling genes and I want to go look at this Wnt signaling gene, and I happen to know that this gene is expressed in the cortical hem. And in these, cortical hem is a very small part of the brain. It's an organizing center for the creation of hippocampus. And I can see a very tiny bit of expression in here for that. Then I could also click on this and click a search for telencephalic vesicle at E13.5. And I would find a number of experiments. I don't have time to go through and mind them all, but you would see that most of these, a lot of these are expressed in the cortical hem. And when I just at a glance, I can see, oh, Wnt, there's other Wnt genes that are highly correlated to Wnt3A. There's a DKK3, we have a Wnt inhibitor, and we have a variety of Wnt activators also showing up in this list. So you can see that all the genes that are really highly marking the cortical hem and highly correlated gene expression pattern to Wnt3A are all Wnt signaling genes. So this is just an interesting way to be able to go through and find candidate genes based upon what you're interested in. So I don't have a lot of time, so I'm going to move on to showing you the AGEA tool. The AGEA tool is here, the Anatomic Gene Expression Atlas. If you'll recall, this is a correlation of the voxel space. So what you have is a seed selector in the left column, and the correlation maps are shown in the other 3 columns. You can choose whatever age you want to do a seed selection. I'm currently on E13.5, and you have 3 planes here that you can, sagittal, coronal, and horizontal. You have 3 planes that you can choose to navigate in to find something that's interesting. You could try to find perhaps something that's interesting in the midbrain, And you can see the correlation maps. Maybe we'll try a part of the thalamus. And what's interesting is you can see how voxels at other ages are also correlated to the voxels at your time point. So basically, I selected this voxel in E13.5. Here's the highest expression. You can see at E15.5, there's some correlation in thalamus to these voxels here as well. So we're doing a correlation across all the ages and all the voxels. So voxels across the entire brain and by age. And then if you want to see the genes that mark that, you click Gene Finder up here at the top, and you have an option to click Find Genes. If you want to be able to see thumbnails of 2 ages of brain, it's nice not to click the Find Genes for the same time point. Click Find Genes at an adjacent time point. And then what happens is you get a list back of all the genes that have a correlation to that expression, that map, that heat map. And you'll get thumbnails over here to the side for each of those time points. So now you'll get a list of genes, and you'll get to see their E13.5 and their E15.5 expression. And you can scroll through them and look for genes that are interesting. So it's a neat way to find genes that, based upon where they might be expressed in the anatomy. And just very quickly, I'm going to show you that if you just wanted a more a search for genes by structure and by age, we have quantified all of the in situ hybridization data. So you can just choose an age, maybe we'll take a P4 here, and choose a region where it is expressed and come up with a list as well. And then you could go through and mine all this data as well. So we had a couple of lessons learned while we were doing this experiment. We thought we were just going to do a quick follow-on to the adult mouse brain atlas and use the same machinery, the same protocols to create this atlas for development. But we found that we ended up having to optimize every single age for the tissue for different problems. We found that we had to use Tyler staging in order to come up with more consistent staging rather than age, because a litter of specimens, a litter of embryos can be at different stages of development in the same litter. We saw anatomic malformations occurring with some frequency. We found that cryosectioning each age was a little bit different based upon the consistency of the tissue, and so it was more of an art than a science. And we had to make a lot of changes to keep delicate tissue adhered to the slide. We had to use a counterstain, HP yellow counterstain, to make the background of the tissue show up, which meant people had to wear a lot of PPE, and the 5-normal hydrochloric acid used in that protocol was a little damaging to some of the equipment. There were a lot of things we had to learn during this process, but we were very excited to come up with this nice standardized set of in situ hybridization across development. So in closing thoughts, common coordinate frameworks for anatomic data that we used in this project, standard requirement in consortia for linking data now. An improved 3D framework is in progress as a collaboration between the Allen Institute's Lydia Ng and Yang-Soo Kim at Penn State. And they will be adapting the reference atlas drawn by Luis Pueyas for our project. And new projects in the scientific community are now doing a lot of additional profiling of brain development with single cell transcriptomics. It'll be really exciting to see that data also coming out into the community for everyone to use. With that, I am going to hand this over to Jeremy. Thank you very much for listening to me today.
Oops. Hi, everyone. Thanks, Carol. I'm going to present the BrainSpan Atlas of the Developing Human Brain. So let me share my screen. Can you see my screen? Great. Okay, so yeah, so that was the mouse developing atlas, and there is a matching human developing atlas. And so why would we want to study human? So while there are a whole bunch of things that are conserved across mammalian species in development, as like Caitlin and Carol talked about, there are a few things that are actually different in primate and human. The first kind of obvious one, if you just look on the left, the bottom is the mouse brain at several prenatal time points. The top one is a monkey brain at roughly matched prenatal time points, and you can see it's just a whole lot bigger for one. In particular, there's this outer subventricular zone, which is important for developing a lot of the neurons that make the cortex bigger, that is pretty large in monkey and human and is almost absent in mouse. There's also this subplate zone, which is much extended in its generation in monkey compared to it being pretty early developed in mouse. There's a subpial granular zone along the top of the sort of the outer surface of the cortex that's largely absent in mouse. There may be some local generation of interneurons in human, although this is controversial. And then just in general, the cortical part of the sort of cortex part of the brain in larger brain species like human are generally bigger than in mouse. So while a lot of things are conserved, there are several things that are different, and when it comes down to it, a lot of the things we want to learn about is in humans, so it's good to have this human atlas. If you go to the main BrainSpan atlas of the developing human brain, which I will show you this later, exactly how you get here. The main page has kind of an overview of this project. This was a large consortium project involving the Allen Institute and several other institutions. It was funded by the NIH, and you can kind of see more about these collaborators on the main page. This atlas includes a whole bunch of resources, in part because of the collaborative nature of it. Three of the main resources are the developmental transcriptome, which is basically focuses on the time aspect of development. There's the prenatal LMD microarray, which focuses on more of the spatial aspect, and then there's the in situ hybridization one, which is the cellular level aspect of gene expression. For the developmental transcriptome, this basically has roughly 50 different donors across development from very early prenatal through sort of middle age, and was used using RNA sequencing and exon arrays. Both RNA sequencing and exon arrays and microarrays are all methods where basically you get a piece of tissue, you put it on a chip, and you get back readouts for the expression levels of a gene for every gene for that one piece of tissue. For RNA sequencing and exon arrays, we can actually get it not just for the gene, but also for either the individual base pairs in the genome or for the exon. So you actually get a little bit more information from this, which is what I mean when I say the transcriptome resolution is high for this atlas. For this atlas, the anatomical resolution is coarse, by which I mean we basically take a chunk of tissue and put it on for each of these different, I think 20 or so brain regions. But we get sort of a good temporal view or time view for genes. Here's an example for an individual gene in the kind of exon level. So these are different exons for SLC17A7. These are kind of their starting positions in the genome and the expression patterns for that exon. And in this case, almost all the exons show roughly the same pattern across space and time. And I'll show another example of this in the demo. For the prenatal LMD microarray, this atlas was done using 4 brains at 2 prenatal, sort of mid prenatal time points across 25 different areas of neocortex, as well as several subcortical structures. And this was done using laser microdissection, which is basically where you take a laser and you cut out a very selected section of tissue that you want. So for example, in say the frontal pole, we cut out 9 different layers in frontal pole as separate microarray values that were run. And this is kind of a blow up of the different anatomical structures or the different layers that were collected. And it's the same one that I showed on the first slide. And so this we have basically gene level values for microarray for a whole bunch of pretty specific regions at two time points. Well, really three, but basically 2 time points. So these are complementary ways of viewing kind of space and time across development. And then the final one of these, and then there's 2 ways of viewing the data, which I will show you in the live demo. One is a heat map, where basically we have just this linear value where each, and it's flipped sideways, but where each, in this case, row represents a single collection point or a single sample. And the values within that row are the expression levels. So green is low and red is high. This left anatomical view is basically showing these values overlaid onto an image of the brain. This is a particular section of the brain that was collected for one of the ages. And so these are basically the same pieces of information shown in 2 different ways. And again, I'll show some examples of this in a little while. The in situ hybridization, the method for collecting it is pretty similar to what Carol said. For prenatal development, we are able to capture a whole section of the brain at once. For postnatal, because the human brain is so big, we have to selectively choose certain sections from certain brain areas. This was the primary sort of atlases for this were these genome-wide ones. And then for a small number of genes that had certain topics of importance, in situ hybridization was also run. And so I'm not going to go through what exactly the reasoning was behind choosing certain genes, but you can see the documentation if you're interested in more details. This table is in that documentation, as well as there's also a white paper that lists exactly the genes that were collected for this. And so again, the advantage of in situ hybridization is you get cellular level resolution. You can see this reelin gene is like right along the edge of the cortex, and when you zoom in, and you can see kind of individual cells that are staining for this one gene. In addition to the gene expression atlases, there are also reference atlases that were done. There were several of them that were built for this project. I think Songlin Dim did at least some of them, if not all of them. There are 3 prenatal atlases, 2 that are of the cerebrum and 1 of the brainstem, and then there's 2 human adult atlases. You can get to these atlases through this link. There's also kind of a more general atlas link that includes all of these, the reference atlas that Carol showed for the developing mouse brain atlas, some adult mouse brain atlases, I think a spinal cord atlas, and a monkey atlas as well. And these are all, at this link, these are all reference atlases. There's also a lot of downloadable information that is available. Most of it's available here. Some of it's available also on the PsycEncode site. This includes imaging, different kinds of imaging from different labs, like structural MRI, diffusion tensor tractography, as well as several other kinds of omics data like methylation and other things. This is just a graphic showing kind of different donors across time and what kind of omics information was collected for them. So there's a whole bunch of information you can download along with the data that goes along with these atlases towards the beginning. So this is what we have on the website. There are some use cases. I'm going to primarily talk about use cases for the gene expression atlases. One, I'm going to go over these first 2 in the demo. The first one is, let's say you're studying autism and there's a particular gene you're interested in, and you want to know when and where it's expressed in development. You can go to the atlas and get that information. So you can look at the cellular resolution of it from ISH through BrainSpan also, and we can see which genes have similar pattern to these. This uses a correlation, find genes like me search, very similar in flavor to what Carol presented earlier. We can also ask about which genes are enriched in different layers of cortex at a particular time point. So I'll show a couple of examples of that. This is something that once we have these lists, we could ask, does it look the same in developing mouse? So you could have a tab open for the human and the mouse, run similar searches and see if you get similar results. We can see how these genes that we find enriched in a particular layer, how they change with development. And we can also ask using our cell types database what cell types they're expressed in, if any, in the adult human or mouse. And then another question that might be of interest is kind of what genes are expressed in the front of the brain versus the back of the brain. And in prenatal development, this is an interesting question because you have both sort of gradients across cortex in space, but also different areas of the cortex develop at different times. So you can find kind of both spatial and temporal gradients doing this kind of a search. I don't think I'm gonna have time to talk about this. And then of course, none of this funding is possible. None of this is possible without funding from Paul Allen, and we thank him for his vision, encouragement, and support. It says thank you for listening, but I'm actually going to go into the live demo, so you have to keep listening for a little bit longer. So okay, so this is the main page right here, brain-map.org. As Carol showed, you can get to this atlas as well through the menu at the top to jump into kind of either the main part or any of the subparts, or by scrolling down to the human brain atlas, developing human brain. And then we click here, it takes you to this main page that I mentioned before. Kind of here's the different consortium members and the funding, and here's kind of a way of jumping into these other databases, which you can also jump in from the tabs up here. So for the first use case that I wanted to go into is, let's say I'm doing an experiment, I'm studying autism. There's a specific autism gene I'm thinking of that I want to look at. How can I find exactly where and when this gene is expressed? So I'm going to start by going to this prenatal laser microdissection microarray, going down to this browse by category and clicking on this autism link. It brings up a whole set of genes that are expressed, that someone had found to be related to autism. When I'm interested in AUTS2, so I'm going to click somewhere on this bar, and it'll tell me information about which structure I clicked on, which also corresponds to this black line here, and some information about the gene that I clicked on. So in this case, AUTS2. Just like with the mouse atlas, I can click over here if I want to and add it to a cart and view just the selections for that gene. So I will do that now. I get AUTS2 now by itself because that was the only one I selected. The way things are organized now is first by donor, which also is by age, which is this top bar, and then by structure, which is this bottom bar. If I want to, I can switch that by clicking these arrows here to kind of reverse those. I want to keep it by age, so I'm going to switch it back. This gene seems to be expressed in kind of a lot of different places, and this linear view is making it hard for me to see exactly which one it is. So if I go up to this gene info and I click on the AUTS2 next to the symbol, it will take me to this other view, this anatomical view that I showed in the slides. This is showing for a particular donor, in this case, the 15 post-conceptual weeks donor, all the sections where microarray data was collected and their expression patterns. So for this particular, for this particular heat map, which is red to blue, or sorry, blue to red, blue is low expression and red is high expression. You can change it if you want to other colors. And so this gene seems to be expressed mostly in cortical structures, and in some of these sort of internal subcortical structures is where it seems to have lower expression. So I can, if I want to look at a different donor, I can change it up here to say the 21 weeks one, and it shows sort of the same sections for that donor here. I'm going to go back now to this view, and great, I found this gene I'm interested in. I want to know which genes also have the same pattern. If I basically click anywhere on AUTS2, go up to the upper right here and click on this Find Correlates button, it shows me a set of other genes that have similar expression patterns to AUTS2. One of the genes in this list is SOX11, which is a key modulator in neurodevelopment, which would give me a hypothesis that would, of course, have to follow up in some other way that maybe AUTS2 is also kind of important in similar processes as SOX11. Okay, so this is just in space for a couple of prenatal time points. What does AUTS2 look like when we look at its expression over time? And to check that, I'm going to open the Developing Transcriptome tab here. I'm going to open it in another tab because I'm going to want to do a comparison in a minute. This brings me to basically a page that looks exactly the same as the one from prenatal LMD microarray. I know that OTS2 is the one I want to look for, so I'm going to type OTS2 in the box. If I spell it right, it's easier to find. And then it brings me to a view that looks very similar to the one from the LMD microarray, except in this case, the donors, as I sort of zoom across, you can see up here, they span a larger age range. And again, I can switch whether it's showing first age and structure or then structure and age by clicking on this box on the right. And just by looking at this, you can say in this case, the heat map is going blue to yellow to red. So we can see that the expression of OTS2 is generally higher in prenatal development and seems to turn off in postnatal development. If I want to see whether this is a gene level, a thing that happens across the whole gene, or whether certain exons are involved, I can once again go to this gene info and click on the gene symbol here. You'll notice the theme here. Typically, if you click on the gene symbol on a page, it will take you to some more information about that gene or other views. In this case, it's showing this view. OTS2 has a whole bunch of different exons, which are shown up here. And sort of corresponding to the ones that are shown down here. You can see a little vertical bar going across here as I scroll over down here. And it looks like not all of the exons in OTS2 are expressed. For example, this one that I'm hovering over here that has the 83 next to it is probably one that's involved in some other organ in the brain and is not expressed in brain, whereas, sorry, some other organ in the body and is not expressed in brain, whereas these down here are ones that have pretty high expression in brain. So this is the advantage you could potentially get from the exon level view, is it's not every exon in the gene showing the same pattern. But for the ones that have expression, it seems that they're all showing roughly the same kind of pattern of high expression in prenatal to lower expression in postnatal. Okay, and so now I'm going to go back, hit the back button to get back to the page showing OTS2. And again, I'm going to click on this Find Correlates button to see which genes have expression pattern, have similar expression patterns across time. And if we look on this list, we see SOX11 again. So probably more than multiple of these genes are going to be similar when we do this search that is sort of a time-focused search, versus when I tab back over to this prenatal LMD microarray, this more spatial search. If we wanted to do a more careful comparison of this, we could scroll down to the bottom of either of these pages and click on the download this data button, and it will let you download sort of anywhere from 1 to 2,000 of the top genes of whatever you happen to be showing in this heat map at the time. So at the moment, this is the top set of genes that are correlated with OTS2. When I originally went in and clicked on the autism genes, it was showing all of the genes in that list. It will let you download whatever you're showing kind of at that time. If you wanted to get all of the data, then you could do that through the download the data button. But this will let you get a kind of a focused view of whatever you happen to be looking at. And you could potentially download sort of these genes, come back over to this tab, download these genes, and do some sort of out of the browser comparison if you wanted to. Okay. Then another use case that we might want to do is you're coming in and you are interested in a particular brain structure. Let's say we're interested in the cortical plate, and we want to see which genes are expressed in cortical plate higher than in other structures. The way you do that is by going over here underneath this gene search button. There's a button that says differential search, where you can get different structures that you're interested in. In this case, I'm interested in comparing the cortical plate expression versus expression. In this case, neural plate covers everything. So this is saying cortical plate versus, versus everything else. And if we click search, it will show the set of genes that have high expression in cortical plate and lower expression kind of in other structures. SATB2, which is one of the top genes, is something that is a common marker of kind of all neurons in adults. FESF2 is a marker for a lot of deep layer cortical cell types. I don't know a lot of these other genes, but if I wanted to, we could go to the Allen cell types database and type these genes in and see kind of what expressions or what cell types are expressed in adult. And remember, the cortical plate is the structure that contains the neurons that end up in the adult brain primarily. Okay. One thing you can also do is this is showing only the genes that are most highly expressed in cortical plate. If we hit this little circle arrow on the right, it'll switch them, and now it'll show you the genes that have the lowest expression in cortical plate. I generally don't use this, but it's something that can be useful if you want to look at both overexpression versus underexpression in certain cases. So kind of for the last thing I'm going to do is now I want to look at cells that are in the ventricular and subventricular zones. You can put multiple things in this search if you want to. These are the germinal zones. So these are the structures that eventually disappear and turn and create some of the neurons in the glia in the adult brain. So if we search these structures, you can see these this big red block. So these are genes that are kind of just high expression in those structures and generally lower expression everywhere. The top hit here is this EOMS, or it's also known as TBR2, and this is a key regulator of neurogenesis that kind of starting from the precursor cells. And so this is a good sanity check that yes, we're getting things we want when the top hit is something that we expect to see. It is also a gene that I happen to know we have in situ hybridization data for. So I'm going to go over to the in situ hybridization tab here, and we can type in this EOMS, and it will take us to a list of kind of all of the experiments that we have this data for. In this case, we have ones for multiple donors across different ages and different sexes across the whole brain, or just in the brainspan or cerebellum. I'm going to click on the 21 post-conceptual weeks 1 for brain. Again, you click on the one under the gene symbol, and it will take you to that experiment. And you can do many of the same kinds of things that you can do in the mouse developing brain atlas. You can zoom in by using the scroll wheel, or by using the scroll wheel, or by using these buttons down here. This changes the contrast, and you can switch from the in situ hybridization view to the expression view. And as we were expecting from the microarray data, there's a band of expression right across here. This covers the ventricular and subventricular zones, and the expression is pretty much off everywhere else. And so, yeah, so that's kind of all I wanted to say about the use cases. Here's the links into the reference atlases. As I mentioned, if you go to one of them, it shows you kind of a very well annotated reference atlas. You can click on the atlas to see kind of on the left here what it is that you're clicking on. If you're interested about a particular thing, particular structure, you can click on the structure name and it'll highlight it on the atlas. And then I guess for the last thing, the downloads, there's stuff you can download about the different atlases I talked about, and then kind of all the extra stuff is down here at the bottom. And if you need help, you can always click on documentation or help. But these are this is all I wanted to talk about for this atlas, and I'm happy to I guess take any questions that haven't already been answered for either me or Carol. Let me stop sharing my screen. All right.
So thank you, both of you. We just have a couple of questions that haven't previously been answered. So those of you who are watching on Zoom, you can click on the Q&A button to read the answers to the questions that we have answered by typing them. And we will also be posting in our community forum the answers to all of our questions, so you can ask follow-up questions on there as well. So Carol, I see you've flagged a question to answer live. Are the strip of thumbnail images always from lateral to medial view?
So for the developing mouse, if you're looking at sagittal images, we do always organize them from lateral to medial. And for the younger time points, it will go past the midpoint of the brain as well and a little bit into the other hemisphere. For coronal, it will always go from anterior to posterior. Jeremy, do you want to comment on human?
I don't want to comment on human. I don't think there's really any thumbnail views to show there.
We have 2 last questions. Is there a reason why the ISH data for the developing human brain versus the other portal, neurotransmitter study, cortex study, et cetera, that's the adult human brain atlas, are not linked? For example, search in the development portal does not find cases that are in the other portal.
As far as I know, that is sort of a practical reason, just that these were separate atlases that were generated at separate times, and the ones that are were put in to different places had kind of different backends that were used. So it's not very practical it would take a lot of work to be able to put the data that was generated in these different systems in the same system. But that is certainly a great idea, having some way of sort of listing all of the genes that are available for ISH from both of the resources.
And then this is the last question we have time for today, which is how can I download the atlases? So if you can both just quickly review where to download the data, that would be great.
So there are a variety of different things you could download or ways to get things. If you're talking about the reference atlases, I believe you can download a view through the API, but I would have to go back and remind myself kind of what that looks like. And I know there's a whole page on the API. Maybe Caitlin could find it quicker than I can, that gives you kind of example queries for how you request different things from the website to download. You can also use that to download images from any of the reference atlas data. So if you actually wanted to download all the in situ hybridization data or the quantification of that information, you can get all of that through our public API. I'm just not able to locate the exact link for that right now on the fly.
And then for human, almost all of the stuff that's available for download is on the download tab. The raw data for the RNA sequencing is, I think it's publicly available somewhere, which I can't remember off the top of my head. I think it's Geo, but you can, we'll put in the Q&A exactly how to get to that on the community forum for people who are interested.
Thank you. Well, we are just about out of time. I do see that we have a couple of remaining questions that are in the chat panel. I have grabbed those. We will write up answers to them and we will share them in a post on our community forum that you can find. We'll link it in the YouTube description. So please come tune back into our YouTube channel and you'll find the link to our community forum post where you can read answers to all of the questions and ask follow-up questions later. Thank you everyone for joining us today. I hope we'll see you at some of our upcoming additional seminars in this series, and have a good rest of your day.